So just maybe two words of introduction about myself. I'm Anders Garm from University of Copenhagen. I'm a zoologist and a neurobiologist. And what interests me during the last 10, 15 years is especially sensory organs, how are sensory organs organized to pick up the exact information any animal need at any given time. I've been working both on chemoreception, mechanoreception, electroreception, but most of my work has been done on photoreception and especially vision. So I've worked quite a bit with eyes, how eyes work and how they differ between animals and how that supports different kind of visually guided behaviors. And what I've become more and more interested in during the last few years is visual processing. What actually happens with the information eyes pick up afterwards in the brain and how that then, of course, in the end turns out to control the behavior. And that's, of course, some of the things I'm going to talk about today. I've also been working with quite a few different animals, crustaceans, echinoderms, annelids. But one of my favorite groups have always been cnidarians for several reasons. And the picture you see here is one of my favorite pets, the Tripedalia cytosphora, which is a small box jellyfish from the Caribbean. And that's going to be the main topic today. There are many reasons to look at cnidarians, I believe, but one of the things I would like to convince you is that they are really cool model animals because in many ways they're pretty simple, hopefully allowing us to understand things in pretty great details. So what I'll do today is first, because I sort of realize that you probably don't know these animals as well as I do. So I will introduce you to the animals, their visual system, some of their visually guided behaviors, and how their nervous system is organized. And then in the end, we will, of course, talk about these learning experiments that we did and published last year. So this is the box jellyfish, and just for you to get the proportions right, this is a pretty small box jellyfish. The diameter of the bill is about 2 centimeters maximum, and the tentacles get about 4 centimeters maximum. So a pretty small species, which is also nice for our work in the lab. But just to make sure that we are on the same page here. So here we have a phylogeny of the cnidarians, the 6 classes that is typically recognized now. The most basal group are the anthozoans. You probably know all the sea anemones and corals here. A small, very strange group called the mixozoas, which are a parasitic group just recently added to the cnidarians. And then we have the major group here, which together we call the medusozoans, of course, because they have a medusa in their life cycle. And in here we find the cubozoans, or the box jellyfish, as a sister group to the more commonly known jellyfish, those you know as the lion's mane, the one we have here, or the moon jelly, which is also a scyphozoan. But the cubozoans here is sister group to those medusae. It's a small class with only about 60 described species. Most of them are in the tropics, but they're also found in the subtropics. These are some of the species we've been working on. And if some of you have heard about box jellyfish before, it's very likely because of this sign you see up here to the right from Northern Australia, where they warn you against marine stingers. And the one they particularly warn you again is this one over here, Chironex fleckeri, which is potentially one of the most venomous animals in the world. At least it on a regular basis kills people in Australia. Their statistics says that 3 meters of tentacle, if that's you get in contact with that, you will have a chance of you will probably die of a heart attack within 2 minutes. So it's a pretty strong nerve toxin. So we have done work on that, but to spare our students, we have been focusing on Tripedalia down here, which, as I said before, is a very small species from the Caribbean, and it's about as venomous as the moon jelly. So it's easy to handle, and we can grow that one in the lab. So it has lots of advantages. So this is the one I'll be focusing on today. There will be data for some other species, but mostly from Tripedalia. But maybe just a few general words about these animals and why so venomous. These are predators, and Chironex is specializing in fish eating. So this here is a Chironex in our tanks in Australia and here in the mangrove. And you see here it has caught a fish. And they basically need this very strong nerve toxin to make sure that when they catch a fish, that it just doesn't rip their tentacles off or destroy the bill. And it's pretty amazing when we feed them a fish, as we've done here in the video, and the fish touches the tentacle. It's two tail strokes, and then it's either dead or stunned. So it's a pretty efficient nerve toxin. So the reason why we're interested in them is not the toxin, but the initial interest came from watching their behavior. So if you look at a typical scyphozoan here, like the moon jelly, they are basically without much behavior. They're planktonic animals just drifting in the currents, and the only thing they really control is a little bit their vertical position. But if you compare them with Tripedalia here from their natural habitat in the Caribbean, you obviously see a huge difference in their behavior. The box jellyfish are much more agile swimmers. They do fast turns, they do directional swimming. And if you sort of forget the shape of the animal, it looks much more like a school of fish than actually jellyfish. And this, of course, caught our attention. How can a jellyfish have this kind of active behavior? And what we pretty quickly realized is that one of the reasons is that they have a much more advanced sensory system, including proper eyes. So if you look at the box jellyfish here, you see these four encircled areas with a white dot in the center. And these white dots are their sensory structures that we call ropalia. And when you blow them up, they look like this. So it's sort of a club-shaped structure with a huge crystal in the distal end. And then we have these six brown areas, which are basically eyes. Depending on how you want to define eyes, the four smaller ones to the side, which comes in pairs, which we call the pit and the slit eyes, they're not image forming. So you can maybe call them more appropriate for ocelli. But the two central ones you have here, which we call the upper and lower lens eye, are proper eyes, basically built like our eyes. As the name already implies, they have a proper lens. They are camera type eyes, so it's a single lens with a single pupil and then a hemisphere retina in the back, as you will see in a minute. So they have four of these ropalia, giving them a total of 24 eyes, of which 8 of them are image forming. So we've been very interested in understanding how these eyes work, how well do they see the world, and of course, what visually guided behaviors do they control, and how is this information processed in jellyfish that, if you look in the textbook, are claimed to be brainless. So that's sort of a problem there for them. So we've done lots of electrophysiology. So just very quick, some basic information about the photoreceptors here. So what we find is that these eyes are slow and colorblind. And already here we sort of can see that the amount of information take up is minimized. So here first, just a slide showing the speed of vision, and you can sort of compare it to the shutter speed of a camera. We operate with a term called flicker fusion frequency. So what we do is we apply a sinusoidal flickering light stimulus while we record from the photoreceptors the output, and then we increase the frequency until the photoreceptors no longer respond. And then what will happen then is that their eyes are basically seeing the visual stimulus as a constant stimulus of the average intensity, like a gray basically. And what we can see here from a night active species where there is, of course, less light, and just like with your camera, then everything becomes slower. So at 1 hertz, they nicely follow the stimulus, but already at 3 hertz, it disappears. So they have a flicker fusion frequency somewhere between 2 and 3 hertz. The two colors here are the upper and lower lens eye. But if we take our day-active Tripedalia, which has more light to operate by, the speed becomes a little faster. So they have a flicker fusion frequency up between 7 and 8 hertz. And just for you to have something to compare with, the fastest eyes in the world are from flying insects. And there we're talking about over 300 hertz in temporal resolution. And the human eyes, we're somewhere in between. And depending on light and how you measure it, our flicker fusion frequency is between 40 and 50 hertz. So it's pretty slow eyes, not taking up much information in the temporal domain. And they're also colorblind. Again, using EFIS, what we see here on the graphs are the spectral sensitivity of the photoreceptors. And what you'll see is there's a single peak on each of the curves here, and that shows that all the photoreceptors in the eyes have the same opsin expressed, having the same photopigment. And this means that they don't see the world in color, they only see intensity contrasts. Uh, and this we've shown here for this is our three different species with Tripedalia again up here to the left. And we've also confirmed these results using other techniques like microspectrophotometry, transcriptomics, and immunocytochemistry. There is a classical thing we find in box jellyfish that is also seen here, is that the exact maximum sensitivity in the spectral domain depends on the habitat. And it's typically such that open water species like Cupula civicisi here, that is out in ocean blue water, is most sensitive to these colors. So we see here a peak sensitivity about 460 nanometers here, which is sort of equal to the blue color of the ocean. And that's, of course, because that's the dominant wavelength in that habitat. But when you go into coastal areas like Tripedalia and Carpsella here, there's a green shift from typically algal growth in the water. It becomes much more greenish. And this we also see in the spectral sensitivity. And we see here a peak sensitivity, typically a little above 500 nanometers, which is this blue-green, which is then dominating in that habitat. So again, nicely matching their habitat and no color information. So again, minimizing the information uptake. But of course, one of the most important things about vision is the spatial resolution. How fine details does the eye resolve of the surroundings? And of course, maybe not surprising, what we find in these eyes is pretty blurry vision. They don't see many details. But interestingly, what we did was when you see the cross section here or longitudinal section of a rupalium, you see they connect to the animal by this stalk here. They have the heavy crystal in the distal end, and then we have the two lens eye sectioned along the midline here. And you see the single spherical lens here looking like basically a fish eye, and then the hemisphere-shaped retina in the back here, bordered by screening pigment, ensuring that light can only enter through the pupil here. And the red lines are indicating our ray tracing, mathematical modeling of how light behaves in these lenses after we have measured the refractive index of these lenses. And what we see, especially in the upper lens eye, is that it's actually a pretty good lens. The image is focused really nicely to a single spot with very little, with a very small blur spot. It's not so well focused in the lower lens eye where the blur spot is somewhat bigger. But what you of course realize is that the image falls way outside the retina. So that of course creates on the retina a huge blur spot. And what that results in is a spatial resolution, depending on where you are in the retina, between 10 and 20 degrees, which again means that they have a really strong, what we call low pass filtering, that only see low spatial frequencies, huge objects, giving them very blurry vision. And again, just to give you something to compare to, your optimum resolution is in your fovea, and it's about a minute of an arc. And that's sort of 0.016 degrees. So they have about 1,000 times worse vision that we have. And just to let you see how that the world appears to them, we of course put them on a roller coaster on Santa Monica Beach and modeled how they would see this. And this is how the human eye resolution would see. And of course, we see lots of details on the roller coaster, in the sky, on the beach and whatnot. But with this blurry vision, all these details are filtered away. So the only thing that sort of makes a contrast on the retina are huge objects. And this, of course, already tells us that it's only large objects they're interested in seeing. And you can fairly ask the question, can you support any visually guided behaviors with this very blurry vision? And luckily enough, you can say both for our research and for the jellyfish, the answer to this is yes, you can actually do quite a bit, even though you have this blurry vision. And some of the things we have shown that they do visually is obstacle avoidance, making sure that they don't bump into objects in the surroundings. They navigate their habitats using vision, and then they also detect large food gatherings using vision. So things can still be done even though you have this poor spatial resolution.
Andres, can I ask you something?
Yeah, of course.
Can you go back 2 slides?
Yes, this one.
One more? I mean, yeah. Why do you think it evolved this way? Why didn't the lens or the retina evolve in a way so that the picture is in focus?
That's a very good question, and we of course asked ourselves the same. Why not just have the same lens focus directly on the retina? So one of the things that actually happens here is, and this I didn't mention, is that what you create, especially in the upper lens eye, and this you also have to realize, you can see that the photoreceptors are oblique. These are very special asymmetric visual fields of each of the photoreceptors. And interestingly enough, those visual fields looks like the visual fields of typical motion detectors in other eyes. So what we think they're doing, we haven't directly proven this, only I mean, just by sort of indicating it, comparing to these motion detectors, is that they can shape the visual field much more precisely in this way by combining this typical way of this special way of blurring with the orientation of the photoreceptors. And this gives special filtering properties to the photoreceptors directly. So you don't need the circuitry to actually get a motion detector. You can have it directly built in to the visual field of a single photoreceptor. So this, as I said before, we haven't directly proven this, but this is the hypothesis that we have.
So, sorry, I probably have like also question about that multiple eyes. So it's
Yeah.
sort of like triangulation, yeah, happening. So it can
You could imagine that they could do that for distance detection, but the idea in having many eyes is also a part of this match filtering. You can sort of also compare it to spider eyes. So by having many eyes, you go from a visual system like in our eyes, which we call multi-purpose eyes. So that means that we have one set of eyes that pick up all visual information. And then we, of course, have an enormous brain that has to sort out that information afterwards, basically 30 billion neurons doing this. But when you have many eyes, what you can do is you can have special purpose eyes. So you can have each eye specialized to pick up only a certain part of the visual world, special part of information. And that takes away a lot of processing power for the brain. And this is extremely important for this system here because they have so few neurons to actually process the information. So by having many eyes, each eye can be specialized for a certain type of information and minimizing the need for processing power.
Thank you.
Continue that question.
Yeah.
That mean that each specialized eyes have a different purpose and a different decision.
Yeah.
What will happen if one eye will see something and make the decision to go to that world,
Yeah.
and the other eye see something else to go to this world?
That's a super interesting question. I will actually address, if you have a little patience, I will address this later on when we come to how the nervous system is organized.
Okay, thank you.
So just to understand the visual world of these animals and their visual guided behaviors, we are just going to show you a little bit about how the habitat looks like. So they're living in the mangrove swamps in the Caribbean, and you probably know how a mangrove is organized. You have these open lagunas here, and then they're bordered by the mangrove trees that stand out with their prop roots into the seawater here. These are the prop roots coming down. And then in between these prop roots, you see these small openings. And these openings is what you see here in the large central image. And that's where you find the jellyfish swimming happily around here, just close to the surface. And you see the prop roots coming down here. And another important thing you see here are all the small wild dots, because that's the reason why they're there. In between the prop roots, you have a high concentration of copepods, which is what they feed on. And they're so efficient at doing this that they catch a copepod every other second. And these copepods are only present under the canopies, not out in the laguna. So if they were out there, they would starve to death. So they have to be in here. So that's their microhabitat is in here in between the roots, which becomes important later. And another thing that is important to understand is a very special organization of these Ropalia, their visual system. And this is how they are orienting them. We, of course, know from ourselves that we have very complex musculature controlling the eyes and giving feedback to the brain, so we know exactly where we pick up information. And this takes quite a lot of brain power to control this. So they do it completely different. So as I said before, they have a heavy crystal in the distal end. And if you look in the literature, it's been called a statocist, basically a gravity sensing device. But that's fraud. They don't do that. It's basically a weight that ensures that no matter how the jellyfish orients in the gravity field, the crystal make the stalk bend such that the ropalium is always upright. So no matter how the jellyfish swims around, it has completely control over the vertical part of the visual field. It doesn't have to bother about where the information is picked up. It's always coming from the same direction in each separate eye. So that means that some of the eyes are looking upwards and some of them are looking downwards. So if we look at the visual fields of these four different visual structures, we have that the pit and the upper lens eye are gazing upwards towards the surface of the water, whereas the slit eye and the lower lens eye are observing the underwater world obliquely downwards. The two eyes we'll be focusing on are the upper and the lower lens eyes, because these are the image-forming ones, and these are the ones we basically understand the best. And we'll start with the lower lens eye down here. And what they do with this one is basically obstacle avoidance. So this here is now not Tripedalia, but chironex swimming in the mangrove in Australia. And they also live in the mangrove swamps, as you can see. And even though they have 0.5 meter long tentacles, they easily navigate here. And you see when they come close to a root, just before touching it, they turn around. So of course, we hypothesized that this obstacle avoidance that is characterized by this 120 to 180 degrees turning with increased pulse rate to swim fast, that this was visually guided. So we tested this in a setup here where we control with artificial obstacles and a slow current here forcing the jellyfish that way, how they respond to different visual scenes. And what you see up in the top one here is that a visible one, I mean, they don't have color vision, so red will basically be black to them. So when they come close to it, as it will now in a second, what you see is, just as we saw in the video before, it will approach it, and just a couple of centimeters before touching it, it will turn around now and swim away. But down here, we have see-through obstacles under the water. And you can see the poor jellyfish here. I apologize for the low contrast in the animal, but there is actually a jellyfish pulsing down here. And even though the mechanoreceptors are actually telling the jellyfish that it's about to destroy its body, but because it can't see the obstacle, it can't avoid it. So it actually keeps on trying to fight its way past this one until it dies. So this clearly shows that this is a visually guided behavior. The lower lens eye is seeing these obstacles and making them able to avoid them before bumping into them. So this gives them a pretty interesting diurnal rhythm. So as I said before, during the day, they swim around in between the pop roots here where the food is, and they feed as long as there's light up, and they use the lower lens eye for these obstacle avoidance behaviors. But the problem is at sunset, it becomes too dark for them to see the roots, and then it's too dangerous to be in there. So what happens is that they sink to the bottom and swims a couple of meters, up to 5-6 meters out into the laguna, where they then anchor themselves to seaweed or seagrass. And another very interesting story is that here they actually display a sleep-like state. And then they lie there motionless all night. And then at sunrise, they sort of wake up and then they resurface. And then they have to find their way back, because out here they would starve. So they need to be able to navigate back between the prop roots. And what we hypothesized was that they actually see, because the mangrove water is pretty murky, so you can't see the roots when you're a meter away. But the air is, of course, clear. So we hypothesized that they actually look out through Snell's window and then see the canopies, and then navigate by the canopies, and then, of course, eventually getting to the roots when you swim towards the canopies. So we tested this with the upper lens eye, and pretty interesting. What we could show was that the visual field of this upper lens eye that is constantly pointing straight upwards due to this heavy crystal is just under 100 degrees. And this matches a very interesting physical phenomenon. You probably heard about this Snell's window. So basically what that says is, from Snell's law, is that when light comes from air and passes through the water surface, it refracts. And this means that the 180 degrees of the terrestrial world, this hemisphere here, is compressed into a cone of exactly just under 100 degrees in the water. So what that means, of course, is that the upper lens eye has a visual field that exactly matches Snell's window. So all the visual information, since they are at the surface, that eye gets is from land. So they actually use this eye to gaze up on land, and then see the canopy and orient towards them. And we showed with behavioral experiments that they can only navigate in the right direction when they have the visual input. If we block the canopy in the morning, they're completely disoriented and can't find the proper direction. So the upper lens eye is actually gazing up on land and see the terrestrial world for this navigation every morning back to their feeding habitat. So we have this rhythm where they every day spend the time in between the prop roots feeding, sleep at night out in the mangrove floor here on the bottom, and then in the morning they rise to the surface, and then they look out through Snell's window and use the upper lens eye to see the canopies and navigate back to their habitat. So one of the questions is, of course, with these behaviors, when you have these relatively complex behaviors, you have to have some kind of central nervous system to control this. So the question is, of course, is there a jelly brain? And if you look in the classical zoological literature, they say no. They say a night herring has this typical nerve net structure, and that's all they have. But when you look closer, that's not true. This nerve net is definitely present, but it's mostly the motor neurons. And it's actually a very ingenious way to control that they have synchronous contractions of the bill so they can swim straight. But the question is, of course, is there a brain, or are they actually brainless? And they do have a pretty well-defined central nervous system, actually. And this here is the model we made of the central nervous system in Tripedalia. So you have this nerve ring that encircles the oral part of the bill. And initially, people thought that might be the central part. But what we find is that it's basically a relay system that allows the four rupalia to communicate with each other. Because the four rupalia each have a pretty complex nervous system that we call the rupalial nervous system. And there should be, that was an image. There should have been an image. Oh yeah, I just have your pictures on top of it. So you see up here, just a part of this rupalial nervous system that lies behind the lens eyes. And here we just use one antibody from a specific neurotransmitter to show just one of the subsystems. So there's several systems in there with pretty complex connectivity. So we hypothesize that it's in the rupalia that the advanced things are happening. And if you look here, this only holds approximately 1,000 neurons. And this is one of the things that makes us very interested in this, because these 1,000 neurons are apparently able to do all the visual processing. And as you will hear later, also able to do associative learning. And we really hope that this simple system allows us to dig into all the details. So we can separate them first in three overall populations just from where they're placed and their morphology. So there's some we call flank neurons, we have some retinal associated neurons, and then we have giant neurons. And what we also find in here that indicates that this is where processing happens is that there is very many neurites and synapses. There are the classical Cnidarian synapse you see down here in the bottom, the bidirectional synapse, where there's not a sort of a predefined post and presynaptic cell. There are synapse vesicles on both sides of the synapse, so they can act as either both of them. But we also find quite a bit, and this is also often forgotten in textbooks, we find quite a bit very normal unidirectional synapses, which is often needed for proper information processing. So we have quite a few of those also where you, of course, have a well-defined pre- and post-synaptic cell. And again, as I said before, we also find that this rupalial nervous system has quite a few subsystems defined by immunostain. And this is just one of them, just to show we have the giant neurons here, and they become important for the learning because they are pacemakers. They are the central pattern generators, you can say. And each time the giant neurons fire, the bell do one swim contraction. And they're part of RF amide positive system here that controls the swim behavior, the swim contraction. And they, of course, connect up here through the stalk up into the ring nerve and thereby to the muscles. And now we come to what happens when two eyes see, you can say, conflicting information. And we don't fully understand this, but we've done some interesting results. And a colleague, Rick Satterley, already back in the 70s, did some really interesting results on this. And what he could show was that these Rupalia, they don't act as parallel brains as you would see it in a computer kind of meaning. They are separate units that inhibit each other. So what he could show was that when one pacemaker fires in one rupalium, it goes up into the ring nerve and then it enters the neighboring rupalia through the stalk, and then it inhibits the activity of the pacemaker in that one. So you can say it in a popular way that what we can show is that at any given moment, it's only one rupalium that controls the animal. The three other rupalia are silenced. So you can say the rupalium that sees the most important at any given time overrules the others and then controls the animal. And of course, that will then change if you can say an immediate danger then appears in the visual field of one of the other rupalia, then that one will increase their pacemaker and overrules the others and then control the animal. So even though they have these four parallel systems, they do not sort of, you can say, they don't collaborate. They sort of overrule each other and then one at a time control the animal.
Can I ask a real quick question?
Yes, of course.
So this brings up some interesting questions, right? So if the two rupalias are seeing two different things, how do they decide which one is important?
Yeah.
And which one to follow? Also, what happens if they are seeing two different things simultaneously? Then I'm assuming they will send off this action potential to each other. Then which one gets inhibited and which one keeps up? So
that's definitely true. We could dig down into this in much more detail and ask these different questions. We have tried this, but it's, at least in our experiments, it's been extremely hard to give equal stimulations to the same rupalia. So we haven't really answered, but I'm sure that it would be possible to sort of confuse the jellyfish in ways that it won't be able to respond in a proper way. But what we see in the mimics we have of the natural habitat is that it seems to work, at least in the way the habitat looks like. So what we see is the way it works. How do they determine what is the most important is basically the strength of the stimulus that they receive will result in a higher fire frequency of these output neurons. And it's the frequency that determines whether it overrules the others or not. That's at least what our data. I will not claim that we have not even near to a complete understanding of this, but this is what our data is saying so far.
Very cool.
So to learning now. So we wanted to ask how much can happen in 1,000 neurons, and at least we think that one of the most complex things we know that happens in nervous system is the ability to learn at different levels, of course. We are, of course, not the first ones that have asked whether cnidarians can learn, and already back in the 50s and 60s, a lot of classical experiments were done, especially on Hydra, which is the classical model system within cnidarians, the freshwater polyp. And here it was very clearly shown that they display the simplest form of learning, like habituation, as this experiment here shows, that just like any other sensory system, basically what happens is that when you repeat stimuli, it stops responding slowly, habituating to the stimulus. And if you give it another type of stimulus, you can show it's not, because it's fatigue, it's not running out of energy. Another stimulus will reactivate the system. So they show this kind of habituation. So that has been known for a long time that cnidarians do this. And actually also pretty early, there were indications of advanced learning in this. There were indication of associative learning in sea anemones, but these experiments had some problems. First of all, they did it as classical conditioning, and the conditioned stimuli was extremely strong electrical shocks they gave them 50 shocks of 32 volts, 5 seconds each, and they didn't look for this, but this very likely implemented lots of damage to the tissue, especially the nerve cells. So that leaves some problems analyzing or understanding the results. And also, as the unconditioned stimulus, they used a bright flash of light, which is indifferent to these sea anemones, so an unnatural stimulus. But still, they could show something that resembled associative learning. So we wanted to look further into this and get more firm proof of how this actually might happen in cnidarians. And to do this, we actually went back to some very early experiments we did more than 10 years ago, where we looked at this again, the obstacle avoidance response. You have Tripedalia here in our behavioral arena. And here we can, of course, change the visual scene. And you see an obstacle avoidance response there. And change how the visual world is. And what we asked back then was, how do they respond to different contrasts? And of course, the contrast is between the gray bar here, which would mimic a prop root, and then a white bar that should mimic the open water between the roots. So how do they respond to these when we change contrast between this? And what we found was very interesting, because what we saw that with increasing contrast, they stayed further and further and further away from the roots until when we had black and white stripes with a contrast of 0.9, they constantly made obstacle avoidances and were centered in the middle of the arena all the time. And what it turned out to be was a very important part of their obstacle avoidance behavior, because in order to do this in a meaningful way, they need to know when to turn. Basically, they had to know the distance to the root. If they turn too early, then they have too little time feeding between the roots. If they turn too late, of course, then they would bump into the root and damage their body. So what they're actually using is the contrast as a proxy for distance. And this is also what this image I showed for is showing, that, of course, it also happens in air, but over very long distances. And that's why mountains are hazy in the distance. But underwater, this decline in contrast happens very fast. So a root close by has a high contrast, whereas roots in the background have low contrast. So this can be used as a proxy for distance. But what we realized was that this also poses a problem for the animal, because this relationship between distance and contrast is nowhere near constant. It changes with the quality of the water. Has it been raining? Is there an algal bloom? Has there been wave actions? So they have to learn every day, how clear is the water today? How is the relationship between contrast and distance today? And that was where we thought, now we have the proper way where we can use natural stimuli to test a natural stimulation where they should be able to learn. So the hypothesis we put up was that they would use a combination of the visual input, basically seeing the contrast of the root, and then mechanical input, stress from unsuccessful avoidances when they have thought the root was further away, but then bumped into it. And combining these two sets of information, which of course makes it associative learning, they would learn that a given day, the contrast is distance relationship and this and that, and then they know when to turn. So this hypothesis we then tested in two different ways. We did it first as behavioral experiments in our arena using naive whole animals. So what we did was we put them into the behavioral arena with a semi-low contrast visual scene with the root mimic having a semi-low contrast to the white open water mimic. And what happens when you put a naive jellyfish in there that hasn't seen this world before is that it would think, due to the low contrast, that the roots are at a safe distance. So initially, they would have a pretty high rate of contrast, of contacts, of bumping into the wall, and they would stay at a pretty close distance. But what we could show that already within 7.5 minutes, the jellyfish learn to stay further and further away, have more and more successful avoidances, and of course, most importantly for the jellyfish, fewer and fewer contacts, fewer and fewer unsuccessful. So this supported our hypothesis that they were actually learning this from these unsuccessful optical avoidances. And what we also tested was that it's not just because they don't swim around so much, because they kept the same swim speed throughout these experiments. So it indicated learning, and actually, to our surprise, it indicated fast learning already in 7.5 minutes. And then we did a couple of control experiments to make sure that our interpretation was right. So we could, in 2 different experiments, take away either the mechanical stimulus or the visual stimulus. So in the mechanical control here, we gave them high contrast, which means that they never touch the wall. And here we see a constant high distance to the wall. We see a constant high rate of successful avoidances and no contacts. Basically, no learning, no change of behavior. So that again supported the hypothesis. And then we did the opposite, where we could take away the visual stimulus by giving them a uniform gray wall. There would be no contrasts, and it had the intensity that was an average between the black and the white bar over here. So when you put a jellyfish in here, it starts bumping into the sides right away. So we have a constant low average distance to the wall, very, very few avoidances, and a constant high collision rate with the wall. No change of behavior, again, no learning. So all of this matched the hypothesis that when they can combine mechanical and visual input, they learn to stay away from these low contrast routes in the experiment. So to verify this, we also wanted to do classical conditioning. And here we took advantage of the fact that we can cut off the eyes along with the rhopalian nervous system just by chopping off them here at the stalk. And we can keep those alive happily for a full day in the lab in seawater. And then we could control the experiments much better. So we had as a of course, now we don't have the jellyfish. Oops, we don't have the jellyfish, so we couldn't see the behavior. But luckily enough, as you saw before, the obstacle avoidance behavior has a neurological fingerprint, and that's the increased swim rate when it turns. So we could record the output from the swim pacemaker as a proxy for the behavior. So we know when that rhopalium is telling the animal, now we want to do the turn. And then we could control the orientation, and then we could present root mimics to the lower lens eye and see when would it initiate the behavior. And what we could then see is that with naive rhopalia, we have 3 different contrasts, 0.2, 0.4, and 0.8. And the naive rhopalia only responded to the highest contrast. And then we did the classical conditioning. And here we gave them only 5 very mild electrical shocks of 100 millivolts, each lasting 50 milliseconds, actually mimic of what we know the output from mechanoreceptors look like. So we try to mimic that a mechanosensor signal is coming from the bell where we suspect the mechanoreceptors are situated, and that then we projected that signal into the stalk nerve here as the stimulus. And what we could show after only 5 of these mild electrical stimulations, they start responding to lower contrast routes. So this again, with this classical conditioning, supported that they are actually able to do this associative learning. But of course, again, we needed to do control experiments. So one of the control experiments we did was no conditioning. So we just let time pass and we pressed the bottom just to make it exact same, but there was no voltage connected. And what we see, of course, is that they still respond to the high contrast, but not to the lower afterwards. So no change in behavior there, no learning. We did see actually a stronger response here afterwards. And why that happened, we cannot explain. But there was some strange change happening over time here. But no response to lower contrast. But what we then also did was do the no correlation test, where the unconditioned and the conditioned stimulus was randomly arranged in time. And then what we could see again was that both the naive and afterwards the animal that have had the stimulation would only respond to the strong stimulation, the high contrast. So all of this all supported the conclusion that these jellyfish actually do advanced learning as associative learning. And what they basically do is that they learn to predict a mechanical stress situation from the visual input and avoid that future stress by turning earlier. And that actually makes it a little more, it's a more advanced kind of associative learning, this what is called operant conditioning. So we were really excited about this. And one thing that is, of course, also really nice is that since we can show that this happens in the Rhopalia after we have dissected them off, it means that the circuitry behind all this must be in the Rhopalia nervous system. So we know that this happens in these 1,000 neurons. So that, of course, makes it very easy now. Not very easy, that's definitely an exaggeration, but it allows us now to pinpoint which part of the nervous system that we should address to try and find the mechanisms behind this. And it was the first time outside bilaterians with conventional brains that we could show this kind of learning definitely. And one thing I personally find a very fascinating result is that depending on, of course, how you consider the comb jellies, the ctenophores, and where they are in evolution, this is debated at the moment, but at least classically, cnidarians is the first group, the earliest group to evolve a nervous system. So what this indicates is that already in the very early evolution of neurons, you would have many of the advanced properties present, such as associative learning might have been present in the very first nervous systems, and might actually have been one of the reasons why there have been such strong selection pressure of keeping the nervous system throughout the animal kingdom afterwards. This, of course, is a postulate. So what we're also doing now is trying to show that other cnidarians with even simpler nervous system than Tripedalia will also display associative learning. So this is one of the way we are now progressing this work. But what I am really most excited about is this, that we can now show that the learning happens in these 1,000 neurons in the Rhopalia. So now we want to hopefully use this as a model system to understand down to the molecular and cell level what exactly happens in a neuron when associative learning is taking place. So the first job that we are in the middle of now is to make a connectome of these 1,000 neurons. So what we're doing is that we are doing serial block phase SEM, where we can have a resolution of 5 nanometers in the XY plane and 30 nanometers in the Z plane. And this gives us 3,200 sections of this Rhopalial nervous system. And this took us 3 weeks to scan and gave us nice 3 terabytes of data. So basically what we want to do now in the connectome is, of course, map out every single neuron. And importantly, with this resolution, we can also map out every single synapse. And we can see whether it's a bidirectional synapse or a unidirectional synapse. So we can basically map out every single, you can say, wiring and every single connection in this here. And what we will then be looking at, of course, is a part of this wiring system where information comes from the lower lens eye, the visual information, and information comes from mechanoreceptors through the stalk. And where this information is integrated in the Rhopalian nervous system, that's at least where we would hypothesize that that is where learning must happen. And once we have pinpointed that circuitry in here, we will go in with molecular methods and intercellular EFIS, and then we will try and pinpoint how do these cells change before and after this learning process, and thereby hopefully get some really nice detailed understanding of these changes that happens during learning. But it's, of course, not in something that is done easily. And even though there's only 1,000 neurons, this is a major work task. So we're hoping that machine learning will allow us to map this very fast. But you probably all know that machine learning is not a time saver until you've done a lot of training trials. So right now I have a very lucky postdoc that is sitting with all these images and are doing all these the training of the machine learning. But hopefully she will have a preliminary connectome sometime next year, and then we will start pinpointing the circuitry behind this learning that we see. But that will be for the future to show if we are successful in this. So I think time is up. So just to summarize here in the end, so what I have hopefully shown you here is that box jellyfish are special because they have much more advanced behaviors than other jellyfish, which was what sort of caught our attention in the beginning, and this is at least partly due to them having a very advanced visual system with 24 eyes, of which there are 4 different types, and 2 of these types are image forming, basically the same type of eyes as we have. But these eyes filter the information strongly. They have low spatial resolution, they are pretty slow and colorblind, but still this information is enough to control different kinds of behaviors. And what I focused on here was the obstacle avoidance behavior and navigating the habitat. And what we also saw that was important for the learning was that during these obstacle avoidance behaviors, they use their contrasts between the root and the surrounding water to as a proxy for the distance. And this makes it important for them to be able to learn this, because that relationship between contrast and distance is not constant. So on a daily basis, they learn by combining mechanical and visual input and associative learning to turn at the proper time. And what we also learn, and which is what we are now focusing on understanding better, is that this central part of Tripedalia CNS, the rupal nervous system, is where this learning takes place. And we now want to understand what happens in the circuitry during learning. And of course, there's been a lot of people involved in doing the work here, and hopefully, or luckily enough, also a bit of money to do it. So, and thanks for your attention. So if there are any more questions, I'm of course happy to discuss this some more.
Yeah, thanks very much. That was super cool. One question with the system in the early embryonic stages, how much plasticity is there? Can you do cut and paste? Can you misexpress genes? Like what is the
Yeah, I would love to be able to give you a very exact answer to this. This is one of the things we look into. We have the unfortunate situation that we can only have these animals reproducing asexually in the culture, so we don't have sexual reproduction, so we don't get embryos. But we are working on that, and what we really want to get going is CRISPR Cas in this system, and try these exact things, and see how plastic is this. We want to look at eye development and try and knock out some of these genes, and once we know the neurons behind this, we definitely also want to look into this. But what we do see is that just by looking at gross morphology of the animal is that there is a huge plasticity. We in the wild often find animals missing 1 or 2 rupalia and at a full-grown stage. So that definitely shows that it's not necessary to have all 4 to survive. They can survive with less. And I think that is a sort of a classical lesson learned from Cnidarian is that this radial symmetry that results in duplications of all the system leads to a lot of redundancy. So a lot of the things are basically there just because the way they grow, they reproduce things in a radial symmetric fashion, but they could in principle easily spare a lot of it.
And in the asexual phase, can you do grafting experiments if you introduce new tissues? Does that work or no?
We have not tried with these ones. This can be done. I don't know if you saw, there was a very cool paper last week on ctenophores in current biology, where they crafted two individuals together and actually make them survive. Of course, a very unnatural situation, but successful. And what we can show is that cnidarians, of course, have extreme regenerational powers, and they can reverse their life cycle, and the cubozoans do it in a pretty spectacular way. They have in their gut what is called gastric filaments, which are these finger-like structures that has a lot of enzyme-producing cells and a lot of nematocysts, and they can detach from the tissue, and then they can swim out through the mouth of the jellyfish if it's not doing well. And then and it looks like a separate animal. It swims around. It looks like it's exploring its surroundings. And once it finds the proper place, it settles and metamorphoses into a polyp. And then that polyp will, of course, at a certain point start to metamorphose and make new jellyfish. And the reason for this is that the polyps are much more hardy than the medusas. So if it's poor conditions, they have a higher survival rate as polyps. But direct crafting we haven't tested.
I, that was a great talk. Thanks. I studied color vision in butterflies for my PhD, and this reminds me a little bit of the caterpillars, which have very simple eyes compared to the ommatidia of the adults.
Yeah.
One cool thing we saw in caterpillars and butterflies is even though it looked like they had monochromatic vision based on their photoreceptors, some of their eyes actually had these filtering pigments that they had over part of their eye. And by filtering the light that was coming in, they could distinguish wavelengths even with only one type of photoreceptor. Yeah. So I'm wondering if you've looked at just something simple like eye shine to see what's coming back out, to try to analyze what's going in.
These do not have eye shine, but we have measured the optical properties using microspectral photometry of all the different kind of tissues, and there are no such pigments anywhere. Just to make an interesting, we're actually just publishing this now. We just submitted the paper. The slit eye, which is this more simple eye, what we just find now. Because just one step back is that what has always intrigued me, and you know this if you worked in insect vision, is that many animals have a huge amount of number of opsin genes, excessive number, and we don't understand why. And even Hydra has 40 opsin genes, even though it doesn't even have eyes. So I've been looking into what is this? Is this redundancy again? Do they just have duplicated these genes and they have hung around for weird reasons? So we're looking at the expression, and in Tripedalia, we find 19 opsins. But every single one of them has a very definitive expression patterns. They're very separate, all of them. And what we found in the slit eye are 4 different opsins. So the slit eye might actually have color vision. And if you know Tom Barton in Sussex, he proposed this very interesting hypothesis about color vision, the evolution of color vision. There was this old theory that it was good because color constancy allows you to recognize objects even though the visual world is changing. But what he suggests is that it's actually a very good way to determine distance because color contrast goes away much faster even than just intensity contrast. So you can, by comparing different photoreceptors with different pigments, you can have a pretty accurate distance determination. And he's shown that the way the circuitry is in zebrafish, it actually indicates that this is very likely the way it originally evolved. And then later on, color came in as a proper information channel. So we're looking into whether that might be the case here, that they use this for distance determination, detection also, or whether it's actually proper color vision. Well, we don't know yet.
I have a continued question about learning. You mentioned that you're doing connectons. What about changing or trying to measure the response time of neurons, individual neuron, as a result of stimulus? Like the Hodgkin-Oxley was from giant squid, so why not the new STDP from jellyfish?
Yes, we're definitely looking into that also. And we will do intercellular recordings once we have seen the circuitry. We think we're pinpointing circuitry. This is one of the things we'll do, and definitely we will also look into if this is one of the parameters that might be variable. Certainly.
It seems like that you have really close to it, because if you have subsystem of different specialists, and you can measure how much inhibition and excitation each one of the subsystem have on the same decision. That's kind of you're almost right there.
Yeah, we hope. I mean, this is some of the things we hope to do. One thing that is of course problematic with all the intercellular electrophysiology is that we're poking a little bit in the darkness here. We can't visualize the cells we're aiming at, and the neurites and the cell bodies are tiny. So the success rate is not high, but we're getting better by the day. But it's definitely some of the things we hope to be able to do in the future.
No, thank you. Cool lecture.
Thanks.
Yeah, great talk. I was curious, do you just check if they have dedicated touch sensors, or do these things basically sense mechanical stimulation directly through like stretching of some of these opsins, for example?
They probably do both, but they do have dedicated receptors. They have ciliary mechanoreceptors evenly distributed in their skin. So we at least our hypothesis that these are the ones that register the bumping into objects.
Okay. And do they do electrosensing with those same? Sometimes the same mechanism can also be used for electrosensing.
Yeah, we do have no evidence of them doing that.
Okay, interesting.
We haven't. I can only say we haven't done very thorough testing, but we don't see any indications in the behavior or in the wild that they would need to do this or have the ability.
Cool, thanks.
I just wanted to make a quick comment based upon the previous part of the discussion about finding all these large number of opsin genes.
Yeah.
And I was reading some time ago that even in humans, there are these opsin genes that are, or opsins that are expressed in all different kinds of tissues, not just on the surface tissues, even in the inside. And there is some hypothesis that opsins might also be acting as thermal sensors in non-visual tissues. So just wanted to make that comment.
Definitely. I mean, they have many functions. That's for sure. And of course, they share in evolutionary history with other GPCR and olfactory receptors. So basically, when you look at the opsin tree, the basal things there are doing different things than vision.